Oh, got it. Recording. Okay. Yes. So as Ajit mentioned, today I'll talk a little bit about our work in spatial genomics, but I'll also cover some of the work that we've been doing in spatial transcriptomics. And probably I don't think this audience needs too much of an introduction of the motivation behind the work, but I just want to mention kind of one interesting thing about the field of genomics, at least how we see it philosophically, is that the last 10 years or maybe 15 years or so has really seen the revolution and maturation of sequencing technologies, right? Going from the ability to measure genomes, right, at scale to discover genetic associations, to now being able to measure transcriptomes and epigenomes, being able to measure that from bulk to going to single cell. And I think that has been a very exciting process for both our understanding about the rules and regulatory biology rules underlying cells, and also promising leads about maybe mechanisms involved in disease. But it's been challenging to bridge that world with, traditional pathology and measurements in tissues. And at the end of the day, I think tissue functions are highly cell non-autonomous. And so this recent really excitement about spatial biology comes from the fact that in part because of the maturation of genomic technologies and the inaccessibility of those technologies to look at pathology and morphology, but also kind of this recent explosion of technologies that's really been able to marry these two worlds. And more often, we're going to see basically the ability to measure genomes and transcriptomes and epigenomes directly within cells and tissues. And that might become the de facto way of performing these measurements in the future, right? And we won't have that trade-off, or at least that's the hope. So that's kind of a philosophical view. And then I think on the transcriptomic side, and I'll go through this really quickly because I'm sure this group is very familiar, we're motivated by problems of tissue organization, in part, for example, how are tissues defined by molecularly defined cell types or molecularly defined epigenomics types, right? And we're lucky because the recent advances in single cell really enable us quite rapidly to be able to assess basically the cell type composition of any tissue and what are the markers and molecular annotations that might define it. And we are starting to begin to also translate that into where those cells fit in place in tissues and what are their interactions. I think the next step beyond that is what are the spatially differential gene expression patterns and how does that relate to our what cell types are and how they function. How do cell type state definitions change as a function of their localization in the tissue, who their neighbors are and who they're interacting with. And then in particular, as I said, we want to, measure those changes, whether in development where cell types are being established and there's cell-cell communication that's important for developmental processes, or in disease where, we want to bridge the world of pathology and understand like why cellular interactions and cell localization is related to cell state. And I'm going to talk to you today about two things. One, I'll quickly explain Slide-seq, which is a technology to address the loss of spatial context when performing single-cell RNA sequencing. And I want to bring this up first because then the later half of my work, of the work presented today, is going to be about an adaptation of this approach for measuring genomes. And so I just want to mention that all the work that we've been doing on Slide-seq has been a joint effort between my group and Evan McCosco's. And the big picture of Slide-seq is effectively lets you do untargeted single cell sequencing. And at the same time, it lets you know where those cells came from in your tissue. And so So how does Slide-seq work in kind of the bigger picture is in Slide-seq we deposit individually barcoded 10 micron polystyrene beads in a monolayer onto a glass surface. And the key is that each bead has millions of oligos on it of this certain format where it has kind of like a PCR handle, a clonal bead barcode, a unique molecular identifier that allows for counting of transcripts, and then a poly-DT capture sequence that captures mRNA. And how this bead barcode works is that it's the same for all the oligos on a bead, but uniquely different for different beads. And because the beads are deposited randomly, we then take a microscope and sequence the clonal bead barcodes for each bead in the array. I won't go into the details of how exactly we do that, but in effect, each base A, G, C, and T and its bead barcode can be captured in one color of a 4-color image, and then we can build up that sequence. And at the end of the day, the XY location of each clonal bead barcode. And so this process of making arrays can be done up front, such that when you perform the experiment, it's relatively easy to perform straightforward. And so you can take fresh frozen tissue and you can section it directly on the array. And then in a very simple protocol, generate sequencing libraries that's very similar to single cell sequencing, including kind of performing the RNA from the tissue is captured locally by the beads, and you perform reverse transcription and then PCR amplification before sequencing. And when you sequence the libraries, you get data that's very much like single cell, in the sense that you end up getting a gene expression matrix, wherein the rows of the matrix are like the individual bead barcodes, and the columns of the matrix are the counts that you observed for each gene across the transcriptome. And now, if you remember, we have the spatial location of each bead barcode, and so you can just match them up, and you end up with kind of a spatial location for each read. Okay, so the big picture then is that you can imagine each bead as like a pixel on a camera. And now you're taking like a picture of your tissue. Instead of like photons, you're like capturing transcripts, and you reconstruct each pixel is then sampling the local transcriptome. So what does the data look like? And this is actually one of our first Slide-seq experiments ever, from a couple years ago, but it explains the point quite well. It has the same data structure as single cell data, so we can apply similar computational tools such as dimensionality reduction and clustering. And here we've ran it on the mouse hippocampus, and on the very left, what we've done is we've clustered the beads and colored them by their molecularly annotated cell type identities. In the middle, there's been no microscopy. Now we've colored each bead by what their molecular annotation is on this TC on the very left. And you can see that recapitulates the tissue architecture in this DAPI stained serial section. But beyond that, now you can assign cell types, such as like, here's the oligodendrocyte layer in between the cortex and hippocampus, and here's CA1 and the dentate granule cells. And so just to summarize maybe a little bit of the specifications and what are important about Slide-seq. The first thing we actually cared about, and one of the important things that we wanted to set out to explore, is that Slide-seq uses 10 micron beads, which are about the size of individual cells. But we wanted to characterize what the spatial resolution of this capture process was. And so we performed kind of single molecule FISH on adjacent sections and measured basically the feature size of genes in single molecule FISH, and compared them with kind of reconstructions in the same physical location on Slide-seq. And we find that basically you get the same measurements at the length scale of single beads. And so we don't think that the diffusion actually is a rate limiting step on the resolution. Instead, you want to consider the resolution is basically dictated by your pixel size, which is 10 microns. And the second thing is that In the last year or two, we really improved the molecular sensitivity of Slide-seq through just a series of enzymatic and molecular biology improvements to something like 1,000 unique molecules per bead. And this is kind of similar to first generation droplet-based single cell approaches. This is and the protocol for how to make these arrays and these things are open source on bioRxiv and protocols.io. And so, and the other thing that we've been doing is we've been demonstrating that like Slide-seq as this poly-DT capture method works across a variety of tissue types, as well as large experiments across many clinical samples and or many serial sections, such as reconstruction of a large volume of tissue and embryos, developing embryos. I won't go into details on those experiments here, but that's some of what we've been focused on in the Slide-seq transcriptomic space. One thing that I do want to highlight is, these sorts of spatial capture transcriptomic methods, Slide-seq being one of them, and I think many of you guys might be familiar with Visium, require novel computational approaches for us really like take advantage of the data. And I showed you that you can just pretend cells pretend each bead is a single cell because Slide-seq has pretty high resolution and cluster it in an unsupervised way. But I think we can do one better, which is that if we have existing single cell RNA-seq references, we can actually decompose each pixel into the constituent cell types and then deconvolve the transcriptomes on each of those pixels. And so we've developed this supervised learning approach to decompose mixtures into single cell types from these spatial capture methods. This is a method called robust cell type decomposition. It works for Slide-seq, it works for Visium, probably works for any sort of pixel-based spatial capture approach. And the idea is pretty simple. If you have a single cell RNA-seq reference, then you want to explain for each pixel on your array what combination of cell types and in what proportion best fit the observed data. This is really work led by Dylan Cable, who's co-advised in my lab, and Rafael Irizarry's lab. And I just want to illustrate why not something like this might be important, which is that in this sort of spatial capture data, even though Slide-seq has quite high resolution, 10 micron resolution, you can end up in situations where each bead captures more than one cell. In fact, we measure, at least in the brain, that each bead captures like the modal number of cells it captures is 1, and the mean number of cells it captures is like 1.5. So 30% of the time you have 2 cells on there. And so you can see that in these 2 cell types for Bergman and Purkinje neurons. These are like interweave cell types in a single layer in the cerebellum. And if you perform unsupervised clustering, here is the spatial plot of beads assigned. You can still see that interweave structure, but if you look at basically the expression of marker genes, either Bergman markers or Purkinje markers, you see that some beads are pretty pure Purkinje and some beads are pretty pure Bergman, and they're assigned by unsupervised clustering to one cluster or the other. But there are many beads which are assigned to one type of cell or the other, but are actually kind of mixes of both cell types. And for maybe many analyses, like let's say we want to do cell type specific spatial differential expression, which is like one thing that we're very interested in. You don't want to say that these mixtures are one cell type or another when they're really a combination of types. And for RCTD, we've performed some detailed computational simulations on which using single cell and single nucleus data sets that demonstrates that we can classify cell types with this method. Here, this is an example where we used like a single nucleus data set as ground truth and used a single cell RNA-seq data set as the reference to classify as the test data set to classify cell types. And you can see that we can accurately classify cell types, but beyond that, using the simulated data, we can mix cell types together in doublets and we can accurately identify them, and we can decompose those into mixtures, into fractional contents of constituent cell types. And this allows us to kind of do 2 different things. One is it allows us to perform really high resolution cell type mapping because oftentimes single cell RNA-seq references are more deeply sequenced. They have more representations of rare cell types. And you don't have like doublets, the problem of doublets. So you can really assign cell types with much higher, I guess, resolution with respect to subtypes. And you can then project those high resolution subtypes into space. Here are just some examples of 27 different interneuron subtypes in the hippocampus. And here are kind of the layered excitatory neuron cell types of the cortex. But I think more excitingly, one thing that we're interested in doing is being able to perform cell type specific differential expression, which means, within a given transcriptomically defined cell type, how does gene expression change as a function of spatial location, right? You see a bunch of variant. If you do single cell, you can see a lot of variance in any given cluster of cells. How much of that variance can you explain by the spatial component of the biology? And here's just like one clear example of that, which is this gene called ALDOC. It's expressed in both Bergman glia, which is that glial cell type that's supporting the Purkinje and Purkinje neurons. And this is performed by a single molecule fish on the same gene. And you can see that in Bergman, this gene is uniform spatially. But in Purkinje neurons, you actually see a very large difference in differential expression between the anterior and posterior part of the cerebellum here. And so this is a spatial difference within one cell type, but not in the other. And this is the sort of stuff that we want to systematically discover. Examples of that might be, so here I just showed you discrete regions, but you can easily imagine that cells, gene expressions might be affected by their neighbors in terms of a cell-cell interaction, and you want to discover that in an unbiased way. Maybe it's affected by pathology, like say that in the case of wounds or plaques or other sorts of pathological conditions. And then another way of thinking about it is that as a function of what cellular microenvironment. I mean, these discrete regions, like this cell type is in different actual, microenvironments. There are different parts of the brain, right? And that might explain this variation. And so we've built a package to do this on top of RCTD for spatial capture transcriptomic data. I won't go exactly into details of how the package works, but it exactly answers those sorts of questions of how do you discover basically gene expression within a cell type, and that varies as a function of where your cell type is. And I want to point out philosophically that's very different than just looking for spatially varying genes, because when you look for spatially varying genes, independent of cell type, you immediately discover marker genes. Like if you go into like Seurat and run the Moran's eye package on your spatial data, you will find the marker genes for the cell types, because cell types are inherently non-uniformly distributed in tissues. If your cells were like completely uniformly distributed and mixed, then you could run spatially varying gene expression, like you could look for spatially varying genes and discover interesting things. But if your cell types are not uniformly distributed, then when you run that sort of analysis, the principal component that you will get out is marker genes. So the first thing is that you have to deconvolve out cell type, and then you have to look at the effects, the residuals, and see like how gene expression patterns are affected by things like interactions or spatial localization. Okay, so now I'll just move on to the spatial genomics part of my talk, which I want to say that one of the main things that we're interested in is figuring out deconvolving gene expression changes, especially in the context of tissue pathology. And one pathological context we're very interested in is cancer. And as I said just now, like we're interested in deconvolving out cell type specific gene expression patterns, right? That's explained by spatial context. in cancer, you have this other component variation is that like tumor cells can have genomic alterations, right? So not only do you want to deconvolve out like the tumor cell, but you might also want to deconvolve out the like cell intrinsic variability, which is like their clonotype or their clonality. And so this is really some work that's been led by Zach Cheng and Tongtan Zhao in mine and Jason Bonroster's lab. And really the technological aspect of it is quite straightforward on top of Slide-seq, is that you can modify these Slide-seq beads. As I said, they're kind of like pixels with where the barcodes kind of delineate where they are in the spatial array. You can modify them with a capture sequence that instead of capturing poly-A RNA, it captures instead DNA fragments which have been transposed with TN5 transposase. And what we can do is that we can take tissue sections and we can transpose the DNA in situ with TN5 transposase, and after that's been the genomic DNA has been transposed, it can actually then be captured by the beads. And we do a protocol which actually eliminates the accessibility sensitivity of TN5 transposase by denaturing the histones. If you don't do that, you can actually generate like accessibility sensitive reads. But for the purpose of this talk, we'll just talk about kind of like genomic DNA sequencing in general. And so one thing I want to point out is that then like these two approaches actually share the bead indexing step. So it's easy to run on serial sections. And so one of the first experiments that we actually ever ran on this sort of spatial DNA seq is we injected metastatic tumor cell clones into mice. These then form like liver metastases tumors. And here's some H&E images of a mouse tumor with this metastasis. And when we run kind of what we call slide DNA seq on it, here, this coloring is, we ran principal components analysis on basically the copy number profile across all of the genome and genomic bins. And the first principal component actually just delineates tumor versus normal cells. And if you look at the loadings, what genomic bins are actually contributing to PC1, then you'll see that basically an amplification in chromosome 6. This arm of chromosome 6 actually explains is one of the loaded parts, but you can actually also see maybe like some other parts of the genome that are lots of parts of the genome here are different in copy number. The resolution of the approach is quite high. Well, I mean, it's lower. I think we think of it in terms of like the resolution of this capture approach is still 10 microns, but we locally aggregate the data to somewhere between 25 and 50 microns to perform the analyses. And but you can see kind of here, this is HMGA2, which is a stain for the tumor cells, and it closely matches in an adjacent section to basically this PC1 signal. And we subsequently injected multiple genotypes of tumor from 2 different tumor cell lines. And now we actually see that PC1, PC2 denote the differences between the different metastatic clones. And if you then look at the loading, you can see that, there's clone-specific chromosomal alterations, right? For example, and these sorts of clone-specific alterations are easily validatable by sequencing the parent tumor cell line. So, chromosome 15 amplification really distinguishes clone B from clone A. And what we actually did subsequently to this is we performed this sort of analysis, slide DNA seq, on a sample of human colorectal adenocarcinoma, and we performed kind of in serial sections H&E and slide DNA-seq, and also, sorry, this is a typo, slide DNA-seq, and then also 10x CNV to get a ground truth of the CNV profiles in this sample. And again, we note that like when we run principal component analysis on the copy number profiles, PC1 denotes the differences between tumor versus non-tumor, and PC2 actually denotes the difference between two tumor clone populations. The difference between those two clonal populations is really probably an amplification on chromosome 20, and chromosome 8q is heavily amplified in all of these cells, and that's actually an amplification in the MYC locus driving these tumors. And here you can actually see an alignment of basically a parallel H&E section, and then tumor cells as identified by slide DNA-seq, and then a serial section where we stain for actual tumors, and you can cells with MKI67, and then the infiltrates in between of normal cells, actually you can see CD45 infiltration. And we actually, as I said, we ran 10x CNV on a very adjacent block of tumor, and you see that you can identify very similar copy number alterations via single cell whole genome sequencing and slide DNA-seq sequencing. Actually, we identify these two clones, which I said were distinguished by chromosome 20 alteration. That's over here, and the green clone has much more enrichment in chromosome 20, and you actually see that in the 10x population as well. Beyond that, you see you can perform a similar computational analysis wherein you can take these clones and project them into the slide DNA-seq data to get higher resolution CNV data. Because we generally tend to have higher coverage in the single cell whole genome sequencing, and here you can identify the same clonal structure, right? So these are clone 2 and this is clone 1, and match them up with the single cell clusters, and now you can actually resolve CNVs up to, I think we have about 10 kilobase resolution in single cell whole genome sequencing, and about 100 kilobase resolution in the slide DNA sequencing. And beyond just running 10x CNV and slide DNA-seq, we actually also ran a serial section of slide RNA-seq, just vanilla Slide-seq. And there we were interested in quantifying kind of these intrinsic versus extrinsic factors which contribute to gene expression in these tumor clusters. And so you can think of one intrinsic factor as the clonotype, right? So like that's like the clonal labels as determined by slide DNA-seq. And an extrinsic factor could be cellular neighborhood. And in fact, we just chose, we were very interested in an extrinsic factor such as like tumor cell density as a function of cellular neighborhood. And we ran a simple analysis, actually, really, a variance decomposition. It's like just like a stepwise regression that decomposed how gene expression within tumor cells as a function of clonal identity, like, you know, like genes which are expressed preferentially in one clone versus another clone, versus cellular neighborhood, like genes which are preferentially expressed in regions of high tumor cell density versus low tumor cell density. So here's like the clonal identities in here. So you can see that for some genes like PLAG1 or FAM3D or MCM7, they have very specific clonal, like PLAG1, 80% of its gene expression can be explained by the clonal identity, right? So that's a cell intrinsic thing. And some of these genes, which are clonally associated, following copy number amplifications, are differential between these two clones. And then you have things which are very cell density explained, but independent of clone. Something like these galactins and prominence turns out if you run gene ontology analysis, many of these density associated gene sets turned out to be basically cell adhesion and coherent molecule classes. So there's like some clone extrinsic program that these cells are running that's affecting their density. And then the subclone associated gene sets, it turns out that there's some differences in MYC targeting that may or may not be. I mean, we think that it should be associated with differences in basically the genomic amplification. So in general, I think that like the combination of spatial genomics and transcriptomics, can really start to deconvolve out like cell intrinsic versus cell extrinsic factors, which we're pretty excited about applying. Yeah, I think just for the interest of time, I won't go too much into the detail for the rest of the talk, but I just want to say that like I made this analogy that Slide-seq is a little bit like converting a sequencer into a microscope, wherein the beads are the pixels. You can really also imagine doing converting the microscope into a sequencer and performing kind of sequencing reactions directly inside of cells. That's another way to do spatial genomics. We have some work in the lab that's looking at much higher resolution, like genome architecture with those approaches. Yeah, so I just want to wrap up here and thank everybody who did the work, including the lab. And here's like the folks who were on the Slide-seq team. And as I mentioned, Zach and Tong and Jason really spearheaded the spatial genomic work.
Thank you very much, Faye. That was a spectacular presentation. We'll move on to questions now. Probably I'll start my questions. I'm pretty sure you get asked this all the time. What about FFPE? Have you tried?
Oh, yeah. I actually think we are definitely working on trying that on. I think the way for FFPE for RNA is probably targeted, and for DNA, I think we could probably just make it work for DNA.
Okay.
I mean, because RNA is very heavily degraded. I think DNA is a lot more stable for FFPE.
Right.
We haven't tried it though for DNA. I mean, I'm pretty confident that you can do untargeted DNA sequencing. Maybe that's something we will try soon.
Yeah, especially in cases like in our lab where we study primary melanoma as a diagnostic necessity, we just don't get fresh samples from patients. Right. So it's all FFP material, and we just
right. Of course. Yeah, no, I think that's a really important challenge, and we're working on it. Hopefully we can have some updates for you soon.
Awesome. Other questions?
I will go ahead if there's no one. Yeah, really exciting talk, very spectacular technology to combine DNA sequencing and RNA sequencing. I'm wondering, for people who would like to give it a try, what would be the next steps? Like, should we reach out to your lab? Because I guess it's not commercially available, obviously, at this point.
Yeah, for sure. I think that would be, I think we're happy. We've been doing some collaborations, mostly in the area, and we've been supporting some other folks by shipping them arrays. So feel free to reach out. I think there is an effort. Oh, and I just disclosed, I mean, there is an effort, like a company has licensed and is interested in commercializing Slide-seq, and so hopefully they can get their act together and distribute it, in which case then.
Right.
That would have been my next question. If you're planning to.
We are not super involved. Like I'm advising them, but maybe it's a little bit of an independent effort on their side. They license the technology and they try to commercialize it. But the goal is on their side to provide it for the community.
Awesome. Probably to rephrase that question, what would be an ideal situation to collaborate with you? Like, what do you expect out of a collaboration in your lab?
I actually think that the experimental part is relatively easy. Maybe there are some people on the call who can actually speak to that. We collaborated with like Jamie Marshall, who's on the call, but actually this computational, excuse me, the computational aspect of it is more challenging in terms of like how, you know, what questions you might want to answer, right? In spatial questions you want to answer. And then like, there are some standardized analyses for like cell type identification or you know, we put out a package for spatial DE, but like, you'd really need to know like a little bit about what questions you want to ask, why it's going to be, why you need spatial like measurements, and you know, have some familiarity with the ability to analyze the data.
Okay, so I know that in your talk you talked a lot about copy number variations. Have you tried going further down for looking at?
Yeah, a single, so like for mutations. Yeah, we're pretty interested in that, and I think that the way to do that is to use targeted enrichment panels, because you're never going to, it's just not cost effective. The libraries are probably diverse enough that you can go and enrich out for variants that you're interested in. But, you know, like if you wanted to do, there's like 10,000 beads, right? Or like 30,000 beads that are capturing cells, and you're not going to do 30,000 genomes worth of sequencing. So I think targeted capture, I think, is going to be important for that.
Other questions?
Hi, Dr. Chen. Very exciting, great presentation for sure. I have one quick question for the last few analysis you showed, the last few slides where you identified intrinsic, I guess, spatial factor affecting some gene distribution. As you, I guess, know, like the 10x, their current spatial transcriptomic platforms seem to be most commercialized at the moment, which suffer from the resolutions.
Right.
Do you foresee any issue for someone trying to apply that to current technology with the 55 micron resolution and do a similar analysis that you've done in those?
Maybe I think it could be done. I mean, we've played a little bit with 10x Visium data as well for spatial, for like RCTD. I think one thing we've observed, at least when you want to do that deconvolution, is that, you know, you lose a lot more power with the larger pixels, at least with the pipelines that we've written, because you have to deconvolve a lot more cell types out, like because like each pixel captures just way more cell types. So that's the main caveat in our hands, but I think it should be doable with similar analysis frameworks.
Okay, yeah. So is that done with the spatial DE?
No, it's like some so we have a package though, actually, that will let you do it. It's called Seaside.
Oh, wait.
I will put it. It's an adaptation of RCTD.
Right. Is that currently called Space XR, the new package?
Oh yeah, Space XR. Oh yeah. I don't understand why we named it this other thing, but sorry, yeah. You're right. Yeah, very good.
Gotcha. Awesome. Yeah.
Here's the package, but the paper is called Seaside, but the package name, I'll ask Dylan about what that is.
Right. We used RCTD before, and I was a little confused when it disappeared and become Space XR. Some of the all the scripts just don't work because they didn't recognize the object, but but yeah, that's great. Thank you.
Oh yes, it's because we wanted a different overall package name that reflected both. Yeah. Mm-hmm.
Any other questions? If not, probably we'll just conclude with one final question. Dr. Fay, thank you very much for coming. But what do you think, what do you see the challenges in the immediate future, and what do you look forward to in the field of spatial transcriptomics and genomics?
I think one challenge, I think the challenge is definitely analysis, because I think collecting data, there's just There's a lot of companies that are commercializing a lot of tech tools right now, and like in the next year or so, it's going to become quite routine to collect data. The question is like, how do you reframe your biological questions into the right computational questions and spatial analysis that also like understands the caveats of like the data that you're collecting from, right? And I think that's certainly been our challenge, and that's both like a challenge and something to be excited about, because I think the exciting thing is that like there's going to be a lot more data out there, right? And people are going to have more accessibility to the technologies.
Yeah, definitely awesome. We have been having a lot of trouble with delineating between human intrinsic events versus extrinsic, like microenvironmental factors influencing the tumor state. And I think Alex Schilek's group in PDAC model using organoids also came out recently showing that genetically similar clones can still have differential tumor state concordance with the microenvironment.
Oh, definitely. Yeah, I think that's really exciting. I mean, we definitely see that, and I think a lot of people see that. The question is like, how do you deconvolve that out?
Right. Yeah. So I'm really looking forward to these technologies being more mature, so that we can all learn more. Thank you so much for coming. It was a spectacular presentation, and thank you for answering our questions.
Of course. Yeah. Thank you guys for having me, and thanks for the great questions.
Thank you. Bye.
Thank you.